Good morning. Let's discuss on heme catabolism. Heme catabolism generally takes place by reticuloendothelial system. Now have look onto the overview of heme catabolism. It is divided in 5 important steps. First is the formation of bilirubin. Second is uptake of bilirubin by the liver. Third is the conjugation of bilirubin in the liver. And fourth is secretion of bilirubin into the bile. Then excretion of, fifth step is excretion of bilirubin. So generally 6 grams hemoglobin is catabolized per day, and the 1 gram of hemoglobin can generate around 35 milligrams of bilirubin. Means nearly 250 to 350 milligrams of bilirubin is produced per day in an adult by the destruction, by the catabolism of 6 gram of hemoglobin. Now move on to the step 1, that is the formation of bilirubin, how it happens. So the reticuloendothelial cells present in the spleen, liver, and bone marrow, they are responsible for the mass destruction of heme. The sources of heme is derived from hemoglobin. So senescent red blood cells, ineffective erythropoiesis product, then degradation of myoglobin and other heme containing proteins. So all of them contribute to the heme ring. This heme ring is going to be broken up, and after being broken up, it is converted up into the biliverdin. So there is a microsomal enzyme system present in the endoplasmic reticulum, and the enzyme is heme oxygenase. And in the presence of oxygen, because it is an oxygenase, NADPH, it converts up into NADP, and this heme is now converted up into the biliverdin. Means the ring, intact ring, porphyrin ring is now broken up, and open ring system is there in the form of biliverdin. So what happens in this particular heme ring? There is cleavage of methynyl bridge between the two pyrrole rings. So pyrrole ring 1 and 2, in between there is a breakage, there is a cleavage, methynyl bridge is broken up and leading to the release of, that's how the methynyl group is detached and released in the form of carbon monoxide. Then uh, finally leading to the opening of heme ring, and then oxidation of ferrous form which is present in the heme. Iron is always present in the ferrous form, so it converts up into the ferric form, and in this ferric form it is now released, which can be recycled further by during the erythropoiesis. Then where biliverdin. So once the biliverdin is being formed in the presence of enzyme, that is the cytosolic enzyme biliverdin reductase, it is converted up into the bilirubin, which is unconjugated, and it is water insoluble. So it's not miscible with blood as well. So here also there is a requirement of 1 molecule of NADPH, which converts up into NAD. So overall 2 NADPH are being oxidized to NADPH. So uptake, step 2 is the uptake of bilirubin by the liver. So once the bilirubin is formed by the reticuloendothelial system, it is now all set to be transported to the liver, but it is being insoluble in blood. So there is a carrier system required for its transportation. So the 1 molecule of albumin can bind to the 2 molecule of bilirubin. Albumin has actually the two binding side, the high affinity side and the lower one is by lower affinity. Via the lower affinity side, it can easily release the bilirubin. To the high affinity side, there is a strong binding of bilirubin to the albumin. So 100 ml of plasma can transport up to 25 milligrams of bilirubin. These figures should be remembered. 100 ml of plasma can transport up to 25 milligrams of bilirubin. So in this carrier mediated form, now it reaches this bilirubin albumin complex, now reaches to the liver via blood, and via active transportation it has to enter into the hepatocyte. So bilirubin enters the hepatocyte via the carrier mediated active transportation. Definitely there is a investment of ATPs, And this bilirubin binds to intracellular proteins which are present in the hepatocytes. The two important protein are liganding protein and Z protein. Liganding protein is glutathione S transferase superfamily, and Z protein belongs to liver type fatty acid binding protein. So these two proteins have important role in intake of bilirubin right from albumin complex to inside of the hepatocyte. So this is how finally the bilirubin is in the hepatocyte. Then the important role of liganding, one is the detachment with albumin and intake into the hepatocyte. Second is these two protein keep the bilirubin in the solubilized form. Then third step is conjugation of bilirubin into the liver. So once in the liver, the bilirubin has to be conjugated because it is completely insoluble and toxic product. So via the xenobiotic system, metabolic system, the it is conjugated into the solubilized form. So it first converted up into the bilirubin monoglucuronide, and finally, it converts up into bilirubin diglucuronide. Now, in this form, it is highly soluble, and this is known as a conjugated protein, conjugated bilirubin. So, we can see the steps. Bilirubin via the enzyme UDP glucuronyl transferase, which is present in the smooth endoplasmic reticulum system, and via the enzyme, via the UDP glucuronate is utilized here, and one UDP is released, and finally the bilirubin monoglucuronide is being formed. In the monoglucuronide form, it is in the nearly 20% concentration. Further, this bilirubin monoglucuronide, which is partially soluble, it is further converted up into the bilirubin diglucuronide by the same enzyme system, UDP glucuronyl transferase. And again, another molecule of UDP glucuronate is utilized here. UDP is released and glucuronate is now conjugated and forming the bilirubin diglucuronide. So 80% conversion of bilirubin happens in the form of diglucuronide. So now it is in the conjugated and soluble form. Next is secretion of bilirubin into the bile. So secretion of bilirubin into the bile also happens by the active transportation system, which is against the concentration gradient from endoplasmic reticulum into the bile canaliculi and then into the bile. And this energy independent process is the rate limiting step. So we can see it here. There is an active transportation and there is an important role of 2 protein again. MRP2 protein, that is the multi-drug resistant associated protein type 2, and CMOAT proteins, they are canalicular multi-specific organic anion transporter protein. So these two important proteins are involved in active transportation of a conjugated bilirubin right from liver to bile canaliculi. So if there is a mutation in these two proteins, in the multi-specific organic anion transporter protein, there are two types of these, two variant of these proteins. So if there are mutations in this protein, that will lead to, inability of bilirubin diglucuronide to be, reached till uh, bile. So that condition is known as Dubin-Johnson syndrome. And this condition, it is an autosomal recessive trait leading to defective excretion of conjugated bilirubin. So the conjugated bilirubin in the blood is increased because from the liver revert back to blood. So the disease results from defective ATP dependent organic anion transport in bile canaliculi. So what will happen? The bilirubin start depositing into the liver and liver appears black. So that condition is also known as black liver jaundice. Right? The plasma bilirubin level seldom rises above 5 to 6 milligrams per day, and patient often have normal aminotransferase level and conjugated hyperbilirubinemia. So this is the picture of Dubin-Johnson syndrome. Then fifth step is excretion of bilirubin. So via bile, this bilirubin now reaches into the gastrointestinal tract, and from here only it is to be excreted out. So we'll see it here. So this is the gallbladder. So via bile, this bilirubin conjugated bilirubin now reaches into the gastrointestinal tract, into the ileum and colon. So in the presence of enzyme, there are a lot of enzymes, bacterial enzymes are present. So beta glucuronidase enzyme, it converts this conjugated bilirubin into the urobilinogen. This urobilinogen, nearly 90% of urobilinogen, it's converted up into the stercobilinogen. Finally, converts up into stercobilin, which is excreted via feces. So nearly 300 milligrams per day of stercobilin is being excreted via feces. Nearly 10 to 20% of urobilinogen is revert back to the blood, and some of it no return back to the liver, and this is through enterohepatic circulation. This is known as enterohepatic circulation, because from intestine back into the liver there is a sort of cycle is being generated. So it comes that bilirubin comes right from liver to intestine, and again from via blood, from blood it again comes to liver. So this cycle is known as enterohepatic circulation. So 10 to 20% of this bilirubin is under the enterohepatic circulation. Then a small amount of urobilinogen, it's been taken up by the kidneys, and from the kidneys, it is excreted via urine in the form of urobilin. And this urobilin gives, a straw yellow color to the urine. So less than 4 milligrams per day is being excreted as urobilin. So this is the overall excretion process of bilirubin, from bilirubin to bilirubin diglucuronide. Finally, via the excretion process, it is excreted out of the body through colon and through kidneys. Now, summary. This is the summary of heme catabolism. So RBC lysis, the broken of RBCs, or those who have completed their 120 days, so they are in the senescent state. They are the sources for hemoglobin, for sources for heme, plus myoglobin degradation, and plus incomplete or insufficient, inefficient heme erythropoiesis, that also becomes the source of heme. Via the reticuloendothelial system, this heme is converted into the bilirubin via biliverdin, and then via bilirubin albumin complex, it reaches to the liver via some active transportation method. This bilirubin enters into the liver hepatocyte. In hepatocyte, they lead to conjugation of this bilirubin, as it is insoluble. So it converts up into the bilirubin diglucuronide, and this bilirubin diglucuronide comes into the small intestine and large intestine in the form of urobilinogen and stercobilinogen. Some of urobilinogen returns back to the blood and enters into the enterohepatic circulation. Little amount is also excreted via the kidneys through urine in the form of urobilin pigment, and the color of urine is straw yellow color of urine is because of presence of urobilin. So all these 5 steps are clearly apparent and explained. Now, what is hyperbilirubinemia? So, a total amount of bilirubin in the blood should be 0.2 to 0.8 milligrams per dl, and conjugated bilirubin used to be 0.0 to 0.2 milligrams per dl. Unconjugated bilirubin should be 0.22 or 0.6 milligrams per dl. This is the normal range. In hyperbilirubinemia, the level of bilirubin should be more than 1 milligrams per dl. And in the latent jaundice, the level is from 1 to 2 milligrams per dl. And the clinical jaundice is associated with when once the bilirubin concentration is more than 2 milligrams per dl. So, these are few inherited and acquired type of hyperbilirubinemia. We will discuss in very brief about all these. First is inherited type and second are acquired. The inherited can be Gilbert syndrome, Crigler-Najjar syndrome, Rotor syndrome, and Dubin-Johnson syndrome. So Gilbert disease, it initially it was believed it is due to the defect in uptake of bilirubin by the liver, but now it is clear there is there is a mutation into the promoter region of UGT1 gene, and this leads to inefficient intake or uptake of bilirubin by the liver. So, so a lot of unconjugated bilirubin starts coming into the blood, raising into the blood. Second is the Crigler-Najjar syndrome. Crigler-Najjar syndrome is of two types, type 1 and type 2. So in type 1, there is a severe deficiency of uridine diphosphate glucuronidase transferase enzyme, which is responsible for the conjugation of bilirubin. And in type 2, there is a mild deficiency of this enzyme, UDP glucuronidase transferase. So the disease is often fatal and the children die before the age of 2. Jaundice usually appears within the first 24 hours of life. Unconjugated bilirubin level increases more than 20 milligrams per dl, and this hence there is a connectiterus. And the type 2 disease is milder form, and the only stage of conjugation is deficient. So this is about the Crigler-Najjar syndrome. Third is the Rotor syndrome. Before Rotor syndrome, we will discuss the Dubin-Johnson syndrome. It is already I have discussed, that is known as black liver jaundice associated with defective excretion of conjugated bilirubin. So conjugated bilirubin in blood is increased. So the disease results from defective ATP dependent organic anion transport in bile canaliculi. ठीक है, already discussed this thing. That condition is known as black liver jaundice because of severe accumulation of conjugated bilirubin into the liver. The next is Rotor syndrome. It is somewhat similar to Dubin-Johnson's syndrome, but the exact defect is not clear. And definitely there is a bilirubin excretion is defective and there is no staining of liver in this condition. That is also autosomal recessive disorder. Then acquired conditions, we will see them more clearly and more comprehensive way when I'll be talking on jaundice. But tentatively, the neonatal jaundice, it can be physiological. So in this condition, it is also known as neonatal hyperbilirubinemia. In all the newborns infant after the second day of life, the mild jaundice appears, and this hyperbilirubinemia is due to the accelerated rate of destruction of RBCs, and also because of the immature hepatic system of conjugation of bilirubin, right? So in all the newborns, the bilirubin level is checked on the third day after the birth, and it is often in the range of 8 to 12 milligrams per dl, and seldom rises above 15 milligrams per dl. So this is phototherapy is being given to such children uh, in order to cope up with the condition. Then three conditions: pre-hepatic, hepatic, and post-hepatic hyperbilirubinemia. So pre-hepatic means there is hemolytic disease of the adult. In this condition, it is characterized by, increased in the level of unconjugated bilirubin into the blood, an absence of bilirubinuria, and excessive excretion of urobilinogen in the urine. And so this is pre-hepatic hyperbilirubinemia or jaundice. Then hepatic can be due to the infective. It can be infective. So most common causes is like viral hepatitis caused by various viruses, hepatitis A, B, C, D, E. So hepatitis virus infection is the cause for hepatic jaundice. So both unconjugated and conjugated type of bilirubin are raised in the blood in this hepatic jaundice. Third is the post-hepatic. So it is mainly the obstructive jaundice, and this is due to the in this condition, the conjugated bilirubin is increased in the blood and it is excreted in the urine. If there is if there is a complete obstruction, so urobilinogen will be decreased in the urine or even sometime absent. So these are the diagnostic there is a diagnostic importance of this urobilinogen in the obstructive jaundice. Right. Due to the obstruction, the conjugated bilirubin is not able to reach into the gastrointestinal tract.